Hi everyone. So we're going to do some two gene crosses for Bio 109, and and I think you can see my whiteboard. There's a problem here that was on one of the practice quizzes. Um, I can't make the text any bigger, so hopefully you can read this. But it says wolves have have a normal coat color, big N, that's dominant over the black, and brown eyes, big B, are dominant over little b. Okay, so I'm going to work on this problem by writing out what I know. So I know that big, so I always tend to write down what I know. So I know that big B is brown and little b is going to be blue. And then N is normal, and then for coat, and then little n is black for coat. Okay, and this other one was for eyes. Okay, and then it says the male is black with blue eyes, and the female is heterozygous. Okay, so she's heterozygous. That's pretty easy. She's going to be big B, big N, little n. And this problem says, what are the gametes for this female? So again, if you have a two gene cross, you look at the the genotype, and you see that there's two B's and there's two N's, and you have to use the FOIL method in order to figure out the gametes. Okay, so remember the FOIL method is first, outer, inner, last. Okay, so we're going to do the first. That means the first of each letter. So that's this one. So that's big B, big N. And then outer means here, the outside letters. So that's big B, little n. And then inner is going to be here. That's little b, big N. And then last, the last of each gene, and that's going to be little b, little n. Okay, so these would be the four gametes for the female. For the male, if you know that he has a black coat, then that means that he had to have little n, and he had blue eyes, so that meant he had little b, right? And so again, if we do the FOIL for him, then we would get little b, little n. And those are and since they're exactly the same, this is the only gamete that is possible for the male. Okay, to double check that, whether or not you did the gametes, whether or not you have gametes, remember gametes are haploid, and so that means that they only have one copy of each gene, so that you should only have one B and one N. Whereas the parents would be diploid, or any babies would also be diploid, and you can tell because they would have two B's and two N's. Okay, hopefully that makes sense. Let's see if we can do the problem. So I don't know if this is actually part of another test question, but let's try to do this. So what if the question was, now you want to mate both this male and this female? Okay, and so you would actually set up the Punnett square, and since I already have it written here, I'll just do that. I'm gonna erase my little thing here. And I'll add in my Punnett square here. Okay, so if I put in my Punnett square, then I actually have the gametes right here, and then I'm bringing in the gametes from the dad. Okay, and then you just basically have a 4 by 1 Punnett square, so it's actually pretty easy. So you could go big B, big N, little n. So again, by convention, you always write the B's first, and you write the capital before the small letter. Okay, and so this is the genotype probabilities of the offspring. And if you want to see, then this one would be obviously brown with a normal coat. This is brown eyes with a black coat. This is blue eyes with a normal coat, and this would be blue eyes with a black coat. Okay, hopefully this makes sense. So I'm going to move on to another problem. So we'll do a new whiteboard. Okay, so we have another problem here, and it says that 70% of Americans get a bitter taste from a chemical called PTC, and this is from a dominant allele that is not being able to, and if you can't taste it, then you have a recessive allele. Then the other thing is that there's another thing called albinism, and that is that it's recessive. Okay, so what I'm going to do is I'm going to write the information down. So I have big A is normal pigment, and little a is albino. Sorry, I think I'll write darker. Okay, and then big T is a taster, and little t is going to be the non-taster. Okay. So again, every time I do a problem, I always write down what I know. And it says a normally pigmented woman who cannot taste. Okay, so I'm just going to try to draw out this little thing. This is a woman, and she is normal pigment, so I know that she has one big A and then something. She cannot taste, so therefore she must be little t, little t. She had a father who was an albino taster, so this is her father, and her father was albino, so he had to have been little a, and he was a taster, so I know that he has one big T and then something, right? But because he was the father, and she got the little T's, one little T from her dad, one little T from her mom, that meant that dad really had a little T there. Okay, the girl also, the woman had one big A, but she had to get something from her dad. The only thing that her dad could give her is a little A. So then that's what we know about this woman. Okay, she marries a guy who is normally pigmented, and so he's got to be normally pigmented, so he's got to be one big A and something. And then he's a taster, so we know he has one big T and something. We know that he has a mother who does not taste PTC, so so she must have been a little t, little t. Okay, so again, that piece of information is just to tell you about the guy. So he was big T, little t. He's homozygous pigmented. So sorry. So he's homozygous pigmented. All right. And so now you need to do the cross. And I would do it. I tend to do them all by the probability method. So here's how I would do it. I do big A, little a from the woman, crossed with the guy who's big A, and I get that 1/2 is big A, and 1/2 is big A, little a. But in reality, all of them have pigment. Okay, so I'm just going to say all big A something. All right. And then for the PTC, you have little t times big T little t. And so then I get 1/2 big T little t, 1/2 little t little t. Okay? And so now I'm going to put them together, and I get, when I put them together now, I'm going to get, when I put them together, then I have to cross all of this with all of that. Because all of these are exactly the same, right? Then that means I will take 1 out of 1 times 1 out of times 1 out of 2, and that gives me 1/2 is going to be big A something and big T little t. And then 1/2 is going to be big A something and then little t. Okay? And that gives you the pigmented taster. And then here, this gives you a pigmented non-taster. Okay, so okay, so I'm doing the same problem but using the Punnett square method, and so again we have the parents, big A, little t, and so you're going to need to figure out the gametes for this parent, and the gametes are going to be big A, little t, and little a, little t, and then this side gametes are going to be big A, big T, and big A, little t. Okay, so here's your Punnett square. So this one is big A, big T, little t, and here's big A, little t. This one is big A, little t, big T, little t, big A, little t, little t, little t. Okay, so again, if you take a look, these two look these two look the same, and these two look the same. I'm sorry, have the same phenotype. Okay, so that would mean these are okay, so these are the non-tasters, and these are the tasters, and everyone has pigment, right? So if you compare this answer with the ones that we did earlier, it will be exactly the same. So and here you have same answer. You get half of them are pigmented tasters and half of them are pigmented non-tasters. So it's the same way, either way you want to do it. Okay, and on the test you can choose to do whichever method that is more comfortable to you. Alrighty.